Okay, so real quick before I start, I just want to thank everyone for coming to my presentation. I really appreciate all of your support, and I appreciate your support, Lila and Angela, as well, as I've gone through this program. Okay. Adaptations in Psychrophiles and Halophiles, an analysis of extremophiles in survival on Europa. A presentation by me, Julia Foringer. So my research question is, how do the adaptations in psychrophilic enzymes provide information on how organisms could survive on Europa? So how I came to this question. I have grown up reading books by Michael Crichton. The Andromeda Strain is my personal favorite, and watching movies like Interstellar. So I've always been really interested in the field of astrobiology. In my free time, I'd go on NASA's astrobiology website and just browse and see what the website had to say about the field. And one of the things that I noticed was that extremophiles played a large role in the field of astrobiology and the search for life on other planets. So going into this program, I knew that I wanted to focus on extremophiles. And as I started the program and did some of my own personal research, I realized that I wanted to focus specifically on psychrophiles because of the adaptations that allow them to survive in freezing temperatures. So originally, I was focusing on psychrophiles and how they could be applied to life on Mars. And I started doing research and finding articles for that topic. However, as I did more research, I noticed that a lot of the data I was finding was very abstract, and I was not going to be able to do a successful analysis on the research that I had found. So I was on vacation at the time, and I think I drew some inspiration from the beach because I turned my focus to an ocean world in our solar system, Europa, which is a moon of Jupiter. And that's how I decided on this research question. So in this presentation, I am going to focus on Europa's environment, horizontal gene transfer in bacteria, and psychrophiles and halophiles and the adaptations found in those organisms. So what are psychrophiles and halophiles? These are two types of organisms that are best suited for survival on Europa. Psychrophiles are organisms that can survive in extremely cold temperatures. They can survive in temperatures from -20 degrees Celsius to 10 degrees Celsius, which in Fahrenheit is from -4 degrees Fahrenheit to 50 degrees Fahrenheit. These organisms are found amongst all three domains of life, which means that they can be bacteria, eukaryotes, or archaea. In addition, psychrophiles are found in the poles of the planet. So the Arctic and the Antarctic, which makes sense because those are the coldest parts of the planet. And like their name implies, these organisms love the freezing temperatures. In addition, halophiles are organisms that can survive in environments that have extremely high salinity levels, from 0.34 molar concentration to a 5.1 molar concentration. And the molar concentration, or the molarity, is just a measure of solutes, or in this case, salts, in water. Halophiles are also found amongst all three domains of life, so they can be bacteria, eukaryotes, or archaea. Like the name implies, the Dead Sea is pretty dead. There's not a lot of organisms that can survive in such a salty body of water. However, there are halophiles that can survive in the Dead Sea. And the Dead Sea is 9.6 times saltier than the ocean. Just to give you a frame of reference for how high of salinities halophiles can survive in. So this picture I have on the slide This is a photo of a culture of Coelia psychrothea, which is a bacteria that is halophilic and psychrophilic. And we're going to learn more about this bacteria later in the presentation. So some background on Europa. Europa is a moon of Jupiter. It has a thick, icy outer shell that is several kilometers thick. And you can see it in this photo. There are some fissures and fractures in this shell as well, which are the lines that you can kind of see crisscrossing the planet. In addition, there is evidence that there is an ocean underneath this icy shell. That's why we call this a subsurface ocean. The evidence for this ocean that NASA has found is that this planet has poles. It has a magnetic polarity that is similar to Earth. In addition, there have been plumes of water found on this planet, and both of these factors imply the existence of this subsurface ocean, which will be important later. Finally, the average temperature on Europa is from negative 160 degrees Celsius to negative 220 degrees Celsius, which in Fahrenheit is from negative 256 degrees Fahrenheit to negative 364 degrees Fahrenheit. So way too cold for me. I'll stick with my warm weather. So my methodology, or how I found the articles I used in this presentation and my literature review. The databases that I used were PubMed and Google Scholar. When I was searching on these databases, I used search terms such as astrobiology and psychrophiles, extremophiles in Europa, psychrophiles and enzymes and astrobiology. Essentially, I hit up all of the combinations in the key words for my research topic. To ensure that my data was valid and reliable, I made sure to only use peer-reviewed articles. And to make sure that my data was current and up to date, I made sure to use articles from 2012 to 2022. So a little bit about my articles. I used 3 literature reviews that focused on Europa. These reviews gave me more information on the geologic processes found on Europa and the moon's environment. I used 1 study in which the researchers used images from a telescope to make an analysis of the planet. I thought that this study was really cool because the authors were from NASA's Jet Propulsion Laboratory, and I want to work for NASA one day, so I loved seeing how the researchers use this information to learn more about the ocean world. I also used 1 model of Europa in which the researchers modeled the moon and used that to learn more about the moon's environment and its geologic processes. I used 6 literature reviews that focused on either halophiles or psychrophiles, or both halophiles and psychrophiles. And these literature reviews gave more information on the adaptations found in these organisms. I also used 3 studies that focused on halophiles and psychrophiles. In these studies, researchers incubated either halophiles or psychrophiles in conditions similar to those on Europa. They then analyzed the growth found in the bacteria to learn more about the adaptations and survival strategies in psychrophiles and halophiles. I did not use any models that focused on either psychrophiles or halophiles. Finally, I used 3 articles that gave me more information on general processes found in cells. I used 2 articles that focused on horizontal gene transfer in bacteria, and I used 1 article that focused on sodium-potassium pumps. I had already learned a lot of this information in AP biology, but I wanted to make sure that I had information to back up the claims I was making. And I did not use any studies or models about these concepts because I felt that I had the best information from the literature reviews. So moving on. My first theme is Europa Application. So on Europa, there is the subsurface ocean and the thick icy shell. However, there is some evidence that these two, this ocean and the shell, interact in a process that is similar to the convection found within the Earth's mantle. So this process takes place within the icy shell. And in this convection, the ice that is closest to the ocean warms up, and then it rises in the shell because it is less dense. At the top of the shell, it cools, and then it falls back down to the bottom of the shell, and the process goes on. So this process implies the existence of oxidation reduction reactions. Oxidation reduction reactions are very important because the redox potential supplies chemical energy for organisms. They can use this chemical energy for the biological reactions that take place. So the reducing agents found within the subsurface ocean and the oxidizing agents found within the shell interact during this convection process to produce oxidation reduction reactions. So the oxidation reduction reactions and the existence of water on this planet implied that Europa could possibly support life. So if this planet could support life, we would probably need to modify an organism so that it could have adaptations and survive on this moon. One way that we could modify an organism is through conjugation. Conjugation is an example of horizontal gene transfer. Horizontal gene transfer is a method through which organisms in a species can share genes and pass on traits that allow them to evolve or better fit in the environment that they're surviving in. So in bacteria, this process can be called conjugation. In conjugation, one bacterium pierces another bacterium and passes on DNA in a circular ring in what we call a plasmid. This new bacterium takes in the DNA and can adapt to its new environment. An organism that we could modify is Coelia psychrothea, as I mentioned on a previous slide. This is an extremophile that is psychrophilic and halophilic, and it is found in deep sea basins. This could be a possible catalyst for survival on Europa, and we could modify this bacteria to have the adaptations it needs. So what are the adaptations this bacteria is going to need to survive on the moon? These adaptations are found in psychrophiles. An example of a psychrophile is Rhodococcus spjg3. This is found in the dry valley of Antarctica, and when incubated in extremely cold temperatures, it was found to have the significant adaptations needed to survive in extremely cold temperatures. So, why do organisms have to adapt to survive in a freezing environment? This is because of their enzymes. Enzymes are made up of amino acids, and they are essential for cellular function. Enzymes catalyze reactions, which means that they can speed them up and help biological reactions in cells take place quickly and efficiently. So, if this is my enzyme, this is what we would call an active site. When an enzyme is catalyzing reactions, a substrate binds to an enzyme. This is where the substrate would go. So If an enzyme is out of its optimal temperature range, it will start to unfold. The optimal temperature range of an enzyme is the range in which the enzyme can function at its best. So if the enzyme is in too cold of temperatures or too hot of temperatures, it gets picky and it will start to unfold. And then its function suffers. We call this unfolding denaturation. So this is my enzyme, this is the active site. And then it gets into too cold of temperatures. It will go through a process that we call cold denaturation, and it will unfold. And then the active site isn't here anymore. So the substrate can't bind to it, which means that the enzyme can't function, and the biological reactions that it was catalyzing will go slower, and the cell will start to suffer. So ways that psychrophiles have adapted within their enzymes to survive freezing temperatures include, well, first, they have less hydrogen bonds within their amino acids, in between their amino acids. Hydrogen bonds are extremely strong bonds found between amino acids, and they are formed between a hydrogen atom and either a nitrogen, oxygen, or fluorine atom. And these bonds hold the amino acids together. So to maintain flexible enzymes, psychrophiles have less of these bonds. In addition, psychrophiles have more beta sheets and alpha helices. So this is kind of what an alpha helix would look like. It's a little spiral. And this is what a beta sheet would look like. But in an actual enzyme, there would be more layers to this. So the enzyme can move within these shapes and maintain flexibility. This way, if it gets into colder temperatures, the enzyme will not unfold as quickly. In addition, enzymes use less of the amino acids arginine and proline, which help them to maintain their flexibility as well. So, next, halophiles also have adaptations that would allow an organism to survive in the subsurface ocean on Europa. An example of a halophile is Quelea psychrothea. It is found in deep sea basins, and when incubated in a brine solution, it showed promising growth that suggests that it could survive on Europa. And as a halophile, it does have adaptations that halophiles use to survive in high salinity environments. So, normal cells that don't have halophilic properties struggle to survive in environments that have a very high salinity level. So why is this? Well, cells have what we call sodium-potassium pumps. These sodium-potassium pumps are located in a cell's membrane, and they use active transport to pump sodium and potassium across the membrane to maintain a chemical gradient of these two salts. An active transport, ATP, which is an energy source, is used to move these molecules across the membrane. However, if the chemical gradient of these salts cannot be maintained and an environment is too salty as compared to the cell, well, water doesn't like this, and the water wants to move out of the cell to maintain an osmotic balance. However, this is not good for the cell, considering that, all organisms need water to survive. So the cell will shrivel up and die, which we don't want. So halophiles have adaptations that allow them to survive in high salinity environments. So these adaptations are very interesting. First, halophiles have extremely regulated membranes, which allow them to stop water from leaving and to help them maintain this chemical gradient. In addition, halophiles will use one of two strategies, either the salt-in strategy or the salt exclusion strategy. And they both sound like opposites, but they both work. First, in the salt-in strategy, the halophile will stockpile and hoard potassium ions within its cytoplasm to help maintain a balance of salts from the environment and within the cell. Or the halophile will use the salt exclusion strategy. And in this strategy, the cell will stop salts from entering the cell. And other than the salts that it needs to survive. So, in conclusion, life can tolerate the freezing temperatures and the high salinity in Europa's subsurface ocean if the organism was able to maintain flexible enzymes like the ones found in psychrophiles, and it would be able to maintain an osmotic balance using the adaptations found in halophiles. And we could modify an organism to survive in these conditions and have the adaptations using the process of conjugation. So, some limitations of my study. A lot of the literature that I found did not address the lack of oxygen on Europa and the possible lack of nutrients and possible radiation found on the planet as well. So if I were to move forward with my research, which hopefully I can, I would want to focus on the pH, radiation, nutrients, and oxygen on Europa and see how that would affect the results I have presented here today. In addition, I would want to turn my focus to Enceladus, which is a moon of Saturn and another ocean world found in our solar system. As humanity expands its reach further and further into the universe, one of our goals is to put life on another planet. And these findings provide more information on how cells found on Earth can give more information towards this goal and towards putting life onto Europa or any other planet in the solar system. So these are some of the sources that I used. One thing I loved about the sources that I found were that I found a lot that had authors from around the world. So I thought that was very cool to see perspectives from different countries and how they viewed this field as well.